Evening guys, welcome to another Sunday session with me, Miss Jackson. Today's topic that we're going to be looking at is paper 2, Biopsychology, as voted by yourselves on the Instagram account. Similar to last week's video, it's just going to be a bit of a brief overview as to what is included within the topic, how we've broken it down booklet by booklet on the spec, and the types of questions that you might get asked about these particular areas. If we do start to find these videos useful, or you would like more kind of in-depth videos on specific parts of the spec, such as numbering questions, looking at past papers, or just kind of a more in-depth revision session, then please let me know on the Instagram story or via email, and I'll kind of have a look at your guys' requests and film some more specific content based on what you need. So if we have a little look at the overview, as you can see on my whiteboard, and I'll pop this image on the Instagram as well. I've just broken it down into the 4 subheadings of the different booklets that we'll look at. I don't know if you can see that relatively clearly. So if we start with booklet 1 is called Biological Structures and Processes. Now, booklet 1 is very much the same as the biological approach, that you've done it already in the approaches topic in paper 2. So in terms of the types of material that's included, this is looking at all of the individual processes and structures within the body and focusing on how our biology impacts our behaviour. That's what's making it more biopsychology as opposed to just generic biology. So for example, you've got things such as the nervous system, evolution, looking at genetics, so the role of twin studies versus adoption studies, MZ, DZ concordance rate, so kind of that general biological knowledge that you will have had to apply into lots of topics throughout the course so far, such as aggression or OCD, or schizophrenia more recently. But then we've also got some new additional content beyond what is in the kind of approaches spec. So on top of the nervous system, synaptic transmission, evolution and genetics, you've also got the addition of looking at the 3 types of neurons, so sensory neuron, motor neuron and relay, and knowing how those 3 neurons kind of fit together. So you need to know the structure and the function of each of those neurons. So you need to be able to kind of draw or label them, if you were lucky enough to get a nice kind of multiple choice or diagram based question. But you also need to know what it is that they actually do. So in terms of the function of each of the neuron, you need to know that sensory is what is detecting that pain or sensation. The relay is assessing that situation and trying to help your brain decide what you're going to do with that information. And then it's the motor neuron that is obviously connected to the muscles and the effector organs, and that is what would have control over your body. So for example, if you stubbed your toe, which is incredibly painful, your sensory neuron would be the one that would detect that sensation. Your relay would make the decision of just how much did that hurt and how are we going to react in response, and it would be your motor neuron that would kind of tell your body, right, quickly move your foot away from the thing you've just stepped on, and kind of allow you to have an appropriate reaction. You've also got the sympathomedullary pathway, often abbreviated to the SAM pathway. So this is looking at in that process of fight or flight. So this ties in directly with what you need to know about the nervous system. In that process of fight or flight, this is looking at that 10-step process of when kind of our peripheral nervous system, so kind of the PNS, is activated and we get to that sympathetic branch of the nervous system where fight or flight occurs. This is looking at how does the hypothalamus in the brain kickstart the SAM pathway, and how does that start to affect our body in terms of producing adrenaline and noradrenaline to prepare us not just physically but also psychologically in terms of focus and concentration. So that's a 10-step process kind of towards the very back of booklet 1, and I would make sure you are familiar with those 10 steps because it's a very common AO1 question to come up, but it's also a very methodical process driven question. So if you know the 10-step bullet points, then you've got everything that you need for that. And then finally, you've also got the addition on booklet 1 of the endocrine system. So this is looking at the role of glands and hormones. My biggest tip for this one would be to remember the difference between a hormone and a neurotransmitter. So neurotransmitters are chemical messengers, particularly within the brain. For example, things like dopamine or serotonin, whereas a hormone is a chemical that travels directly through the bloodstream as opposed to the brain, and it is released from glands that secrete it, and it travels to different target sites around the body. So for example, in the endocrine system, there's lots of glands and hormones that you can actually get credit for in the exam. The main ones that we've taught you in the booklets are the ovaries, which produces obviously estrogen. You've got the testes producing testosterone. You've got the thyroid gland, and we also talk about the role of the adrenal glands, obviously producing adrenaline. So you've got kind of some of the main glands there, in terms of what you would include in an answer. You can actually also get credit for talking about glands such as the pineal gland, which ties in with what I'm going to recap on biological rhythms, in terms of secreting that melatonin around the body and helping with the function of sleeping and waking. But they're kind of the additional elements to booklet 1 that I would be mindful of and make sure you go back over. On booklet 2, this was kind of the chunkiest booklet of the topic, and booklet 2 is all about the brain. So as you can see, it's split up into kind of 3 or 4 sections, but really I like to see this as a booklet of 2 halves. The first 2 sections of the booklet are all about the healthy brain, the neurotypical brain, and kind of when the brain is functioning at its regular capacity, what are the different parts, how do the brain work, either holistically together or separately. And then the second part of the booklet is looking at the brain in response to kind of damage and how it has the ability to recover itself. So the first section of the booklet is split into 2. You've got localization of function, sometimes called cortical specialization, and then you've got lateralization. The way to remember the difference between these is to focus on that word localization, meaning location. So I always think localization, it's about the locations in your brain, so the individual parts and components. And what you've got to know about the 6 main cortexes is you've got to know where they are in the brain, so what lobe and hemisphere are these locations located, and you need to know their function, so what actual function does that have and how does that impact our body and our behaviour, what is it responsible for, and then finally you need to know about what we call aphasia, which is the key term for damage. So what could possibly happen if that part of the brain was damaged? How would that affect your behaviour? Again, it's biopsychology, so linking it back to how behaviour is impacted. So for localization, if you remember the kind of acronym we gave you of brainy mice seem very weirdly aggressive, that stands for Broca's, motor, somatosensory, visual, Wernicke's and auditory. So you need to kind of know whereabouts they're located in the brain. And also if you remember that acronym, that kind of goes round in order from the kind of the frontal lobe, where you've got Broca's at the front of frontal and motor towards the back of frontal lobe, all the way through to Wernicke's and auditory, which are in the temporal lobe at the bottom. So if you remember that acronym and kind of remember that visual, that's really useful if you get like a multiple choice diagram label. You just remember the acronym and it kind of goes round the diagram all in a circle. In terms of lateralization, this is about the individual hemispheres. So what is the function of the entire left hemisphere versus the right? How do they work both collectively via things like the corpus callosum, but also what are their functions separately? So the left, for example, is lateralized for language. The right is more lateralized for recognition. Along with that, that's where you would possibly get questions or bring in your knowledge of Sperry's research into split brain patients. That's kind of a really big piece of research that you would include in a question on lateralization. So it's about remembering the question, really carefully familiarising yourself with the terminology, because it's often the terminology that trips us up. And there was a question on Dep 10, I think it was, or the last assessment where you were assessed on biopsychology alongside research methods, and the 16 mark on that last assessment for biopsych was kind of a localization and or lateralization question. So often you do kind of get asked about them together, but also therefore you need to be able to differentiate between them. In that second part of the booklet I mentioned, you've got what we call brain plasticity and functional recovery. So brain plasticity is this umbrella term for change. All of us experience, whether we are kind of neurotypical, neurodivergent, whether we are kind of somebody with a healthy brain or somebody with a disordered brain, for example, that has been kind of damaged through illness or injury. Everybody goes through brain plasticity. It's just about change. So over time, with age and experience, the more that we use a certain skill, those neural connections will strengthen. But then over time, it's that use it or lose it rule. So any neural connections that we're not particularly using, our brain almost prunes and deletes through a process of what we call synaptic pruning. And that is how we make way for new learning, by getting rid of the neural connections that we no longer need, and kind of making space for that more grey matter. So everybody goes through kind of synaptic pruning as a process of growth and learning. However, functional recovery is a type of brain plasticity, particularly in response to trauma. So through illness or injury, for example, if you were to have a stroke or kind of a brain injury, functional recovery is all about the brain's ability to repair itself to a certain degree through things like neural reorganisation, neural regeneration, axonal sprouting, to create and strengthen new neural connections. Booklet 3 really nicely maps on from that material about the brain, because booklet 3 is called Ways of Investigating the Brain. So this is actually about how we measure brain activity, and looking at the pros and cons of using different methodology and techniques in psychology and in neuroscience to be able to investigate these localized and lateralized areas. So the 4 main ways of measuring the brain that we look at on the specification is an fMRI, so functional magnetic resonance imaging, an EEG, so electroencephalogram, an ERP or an ERP, and that's an event-related potential, and then you've got a post-mortem examination. So your post-mortem is kind of the most obvious and a little bit of the odd one out, because that is a physical examination of the brain after death. The main exam tip with a post-mortem is to remember the purpose of a post-mortem is not to determine cause of death. We're not coroners, we're not on CSI, or trying to figure out why somebody might have passed. The purpose for a psychologist or a neuroscientist doing a post-mortem would be to look at the physical structure of a neurotypical brain versus a neurodivergent one, to look at the differences. Is there anything different neurochemically or structurally in a healthy brain compared to a disordered one, for example, that would allow us to look for any sort of physical connections to that particular behaviour. So it's looking at what are the issues physically that could have maybe explained some of the problems an individual experienced when they were still alive. That used to be very common, for example, in looking at neural correlates in schizophrenia, so things like enlarged ventricles in the brain. Post-mortem used to be kind of the main way that we would identify such differences. But now, with the kind of combination of cognitive neuroscience, we have kind of moved to more of a technology scanning-based method. And the 3 main scanning techniques I've already mentioned is the fMRI, the EEG, the ERP. In terms of this booklet, because it is quite small, the main thing you need to know is kind of the few key bullet points of the features of how each of these methods work, but also mainly being able to compare them. So talking about not just the strengths and weaknesses of each one in terms of how effective or appropriate they are as a way of measuring the brain, but also how suitable are they for determining certain activity, and are certain techniques better than others. So for example, the fMRI is a little bit different in the fact that it uses kind of looking at the magnetic quality in the blood, so kind of identifying and determining how much oxygen is being used in different parts of the brain when you are engaged in a cognitive activity, so it can kind of pinpoint the location. Whereas the EEG and the ERP use electrodes on a skull cap, so that's very much looking more at the electrical impulses by our neurotransmitters and more kind of general brain activity. So the fMRI, for example, is a lot more what we call accurate in terms of spatial resolution. So the two main key terms I would make sure that you are familiar with or that you know about these techniques is whether or not they have good spatial or temporal resolution. So if you break that word down, spatial, think about the space, the accuracy in terms of precision. So how accurate is the scanning technique in determining the exact location the activity is coming from, so kind of to the nearest millimetre, how well can it detect it. Whereas temporal resolution, think about that word temporal. We use it all the time. We talk about temporal validity, for example. So that's about time, okay? It's about how accurate is it in terms of quickness. How immediately can the activity actually be detected, again to the nearest kind of millisecond. So the spatial resolution for the fMRI is really good. It's within kind of 1 to 2 millimeters of accuracy, whereas the EEG and the ERP is a lot more kind of superficial or general brain areas. Whereas the EEG and the ERP, because it works on electrical activity rather than looking at the magnetic quality of the blood, it has a really good temporal resolution. In fact, EEGs and ERPs can detect brain activity in milliseconds, in almost real time as it's happening during a cognitive task, but it's not as accurate spatially. So there's pros and cons of using every method, and it's about being familiar with the key terminology and knowing the specifics about each of those different kind of ways of measuring the brain, including things such as the type of brainwave that they look at and in what context they might be used. You know, is it invasive or not? So is it safe? They're kind of good evaluative points to consider. And then finally, booklet 4 is about biological rhythms. So this is about identifying the 3 main types of bodily rhythms that we have, understanding how that would affect us psychologically and physically in terms of our behaviour, and then being able to use some specific kind of key terms and key studies to help explain how this all works together. So the 3 main biological rhythms that you look at on the spec is a circadian rhythm, an ultradian rhythm and an infradian rhythm. And each of these biological rhythms have kind of different lengths in terms of how long they last and how often they repeat. And the main things that you need to know is not only how long they are and to be able to give a contextualised example, along with maybe some research evidence. It's about knowing what the exogenous zeitgebers and the endogenous pacemakers are for each of those rhythms. Now that seems like a really big word, but basically the endogenous pacemaker is an internal factor, so something physically within us that could govern or change or regulate your biological rhythm. So I remember that in terms of the word pacemaker. So if somebody gets a pacemaker fitted in their heart, that's internal, that's something physical within you. Whereas exogenous zeitgeber, I think exo like exit, so it's something external, it's outside of the body. And also exogenous zeitgeber is German for time giver. And one of the examples of an exogenous zeitgeber is things such as social cues and time. So your circadian rhythm, the main example, and this is actually named on the specification, so for circadian rhythm you have to use this key example really, and that is the sleep-wake cycle. So this is a bodily rhythm that repeats every 24 hours, because within a 24-hour day we have periods of being awake and periods of being asleep. And this is all about how the external factors of things like light and social cues, like getting up for work or for college, how that interacts with our internal mechanisms like the suprachiasmatic nucleus and the pineal gland, to allow us to have that period of feeling awake and feeling tired and wanting to go to sleep, to help regulate our body. And there's some really good key research studies that I would make sure you're revising for circadian rhythms. So the case study of Mikhail Sifra, looking at De Courcy's study with the chipmunks, or Campbell and Murphy. So there's a couple of key studies there that I would make sure you are familiar with, particularly as this is likely to possibly come up as a 16-mark essay. Slightly less commonly asked about is the infradian and the ultradian rhythms. So an infradian rhythm, the cycle itself is longer than 24 hours, but it happens less frequently. So I remember it as infradian is infrequent. So the key example for this one is the menstrual cycle, because it repeats once every 28 days, hopefully. And then you've got the ultradian rhythm. So think ultra, think a lot. This is a rhythm that is quite short, but it happens multiple times. So the actual cycle itself is less than 24 hours. A really good example is the stages of sleep. Be careful not to call it sleep-wake cycle, because that is different. That is a circadian rhythm. The stages of sleep is the 90-minute cycles that occur and repeat throughout the night when you are asleep. So every 90 minutes, your body goes through 5 stages of sleep, kind of looking from stage 1 and 2, where you've got quite light sleep, all the way through to your kind of non-REM sleep, where you are dreaming. So that kind of goes through those 5 stages, and at each stage you've got different brain activities and kind of different functions and purposes. Kind of that very restorative slow wave sleep, where your brain does a lot of cognitive repair and memories are consolidated. So that's kind of also your reminder to get lots of sleep in the run up to exams, because it's essential for helping your memory. But that cycle itself is less than 24 hours, but it does repeat quite often. And that's kind of it for biopsych. So it's giving you a bit of a kind of 15-20 minute rundown of the topic with a few kind of exam hints along the way. Hopefully you found that really useful. Again, I'll do a vote on the Instagram story. I'm possibly thinking maybe approaches overview next week. But let me know what you need. Let me know about other alternative videos you might be interested, especially if you want some more kind of skills focused videos, then I'd be happy to get some of those filmed and get them on the channel. So thanks so much, guys. Have a lovely rest of your evening, and see you back in college tomorrow.